Hello and welcome to BoneUP, the podcast all about bones. I'm David Armstrong.
Hi, and I'm Richie Abel, and we're going to bring you the latest in bone research. So whether you are a patient living with a bone disease, a family member, or a healthcare professional, you've come to the right place.
If you want to learn more about your infrastructure of calcified collagen, this is BoneUP. Hello and welcome back to another very special episode of BoneUp. We're live here at the European Calcified Tissue Society meeting in Girona in northern Spain, and I hope in this episode we bring you another flavour of the wide range of talks and presentations and items for discussion that we enjoyed today.
The listeners, we've been able to track down a really excellent guest today, Marc McKee. Thank you for joining us on the podcast.
My pleasure.
So ECTS is a very varied conference. We have people from all over the world and all different backgrounds, and you have a very interesting background and story. How did you get into bone?
Well, I trained as a cell biologist, and I had an instructor in my final year of undergrad university at McGill who was studying bones and teeth, and I just got entranced by the way that biomineralization, that's the name of the field I work in the broadest sense. Biomineralization uses the mineral ions and rocks that you might see on a walk or a hike on our planet. You pick one of those up, and so those are rocks, and biology has evolved to use the same elements as you would find geologically on a hike. And so what biology has done, it uses what it has available to it, the ions, calcium, phosphorus, for example. Biology harnesses and brings together those same atoms to make hardened skeletons, hardened dentitions, teeth. And I was always fascinated at this convergence point of geology and mineralogy with biology. How does biology integrate biorocks into the body as bones, teeth, cartilage, inner ear otoconia, and so on?
And you've worked on a lot of different biomineralized tissues in your career. Could you tell us about some of them?
Yeah, actually, I'm fascinated by anything that mineralizes or calcifies. We say calcifies because they typically involve calcium, but there are many other types of mineral. And so it always intrigued me, as I told you, how do you incorporate that normally into a physiologically into a vertebrate skeleton, not just humans, but all skeletons? How do you incorporate that into the teeth? Where do those crystals, those nanocrystals of mineral, biorocks, where do they form? Are they forming inside the cells, in fibrous matrices outside the cells, or whatnot? So in different model systems that we look at, different organisms, different structures, different tissues, as we call them, we look very carefully at how and where tiny little nanocrystals of calcium and phosphate or calcium and carbonate, like in avian eggshells, where they're deposited, how they're regulated, just like any cell pathway signaling would be regulated. It has to be very carefully regulated by cells and by proteins, or we would just turn like Lot's wife into pillars of salt. And so how do we regulate that process? It's always fascinated me.
At the conference yesterday, you gave a really excellent talk. It was standing room only at the back. And you were talking about cell culture. Could you describe some of the research to us?
Yeah, so cell cultures, we've had talks on the meeting here about that. Cell cultures are useful for modeling certain things, not all things about biology, which of course is extraordinarily complex. So we can answer some primitive questions using cells in petri dishes, so to speak, in various types of cultures. And most of cell culture work that's been done to date on biomineralization has been using bone cells. And they grow in petri dishes, but usually they just make little nodular spots of mineralized bone-like material in a dish. But we've come to a point where some cells have been isolated and expanded that you can grow from the dish surface up. You can grow three-dimensional structures that resemble this fractal-like growth of trabecular bone that you find in mammalian bone, including in humans. And so we're starting to reach a point where cell cultures are becoming 3D. They're developing three-dimensional structures that are bone-like. And then other people are developing organoid cultures and bone on a chip, this sort of thing. But by doing so, we can answer specific questions. And it's very important in that it can help reduce animal testing and animal use, which we all want to do, in fact. Secondly, they can be used for pre-screening of drugs to see if they might work on these little mini bones in a dish before you use some animal testing, if you're going that way. And thirdly, you can do some gene editing in these cultures. These are living systems, cells and extracellular matrix, collagen fibers, and mineral. And you can, before you make a transgenic mice that can help you understand fundamental principles, you can very quickly and easily just test your gene editing in a petri dish before you even think about involving an animal or human use.
Presumably, bone tissue, more importantly than any other, is perhaps ideal to have a 3D model because bone is load-bearing and weight-bearing. That's the point of bone. So you would imagine a 3D model would be particularly important to replicate what it's actually like in vivo as opposed to just having lying it flat on a Petri dish.
Right. I think that I would prefer analyzing, measuring, understanding the real tissue first, and then work backwards to these cell culture systems that I just mentioned and do some things that we can do with them specifically, like screening, co-cultures, drug development, and so on. But I think the two inform each other, and each little piece of the research puzzle gives you clues that move science forward. So I like to work in vivo, and I do work with research animals, mostly mice, because you have very important ways to manipulate the mice with genetic mutations, and these are called transgenic mice. But there are also spontaneous mutant mice that just develop normally that you can use. So we try and use everything, but at the end of the day, we're trying, or I am trying, to understand how biorocks integrate to give mechanical properties to our skeletons, to our teeth, and our other mineralized tissue that everybody forgets about is our inner ear autoconia that give us sense of acceleration and balance. It's a different type of mineral, but it's another mineral, another biorock that we have in our bodies. So we're trying to understand basic structure by all the tools and instruments and electron microscopes which you need for nanostructure. And we're trying to see how small calcium phosphate crystals in bone, let's say, integrate with fibrous collagen extracellular matrix molecules and the spaces in between to impart the mechanical properties to bones that they have. And so we need to understand structure. We call this structural biology. The structural biology of bone means understanding bone structure at all hierarchical levels. It's a fractal hierarchical tissue. And unless you know each of those levels, you will not understand how it all comes together to give it bone its mechanical properties. And Richie, that's your quest to learn how that works.
That's how I came across your work, the work around tessellation of bone mineral crystals, and I found it really, really fascinating. And really it's opened up whole new fields of research for us, helping us to understand data about bone biomechanics, etc. You had a few slides about the tessellating mineral crystals in bone. Would you tell our listeners about that?
Okay, so yes, we discovered this three-dimensional packaging of bone mineral in that rich collagenous matrix in 3D, and we were able to quantify it. But quite actually, if you peruse the previous literature, going all the way back to Alan Boyd in London, no less, where you're from, if you peruse scanning electron microscopes, where you can see microscopic structure at different scales, there are quite a few reports in the literature of single fractured open bone that's filled with these prolate ellipsoid, little rugby balls shapes, and these are what we call the tessels, tiling. So like the Alhambra in Granada, they're tiling that I'm talking about of the walls, but I'm talking about three-dimensional tiling now. So a bunch of people had seen these tessels, these rugby footballs packing together in bone, and there are some descriptions in them. What we did to give the full breadth of what that means, we were able, working with electron microscope companies who make a new instrument, well, not so new anymore, but a scanning electron microscope instrument called a focused ion beam scanning electron microscope. It's called FIBSEM for short. And what that microscope lets you do is it lets you slice by slice, like a microtome or cutting a piece of cheese, a chunk of cheese, slice by slice, you cut, image, cut off another piece, image the block face, cut off another image, image the bone face, and so on. And you gather together thousands of sections at electron microscope resolution, just like confocal microscopy does the same thing at the light microscopy level. We can now do electron microscope 3D tomography. So we've gone beyond those reported initial images, and we are now doing the slice and view, reconstructing by X-ray tomography, reconstructing those thousands of images and making a three-dimensional volume that you can now quantify. You could never quantify just looking at the surface of something. You don't know what's underneath it. But we could march right through it now in 3D and collect thousands of these tessels, not just a few decorating the surface. We measured and quantified over 2,000 packages of nanocrystals that form tessels, a tiling, a three-dimensional tiling that fill bone. And it's a micro-scale organization of mineral that's been overlooked over the years. People know about the nanocrystals, they know about bone lamellae and bigger structures, but they did not know that there was this organized packaging of 3D tiling, tessellation in bones.
And fundamentally, this is one of the most important things for providing tissue with high strength. And it's going to be critical, I think, in the future, understanding these structures and how they function under load to try and understand why bone is so strong, why it's so fracture resistant at the tissue level.
That's right.
So you've also done some research around rare bone diseases, which in terms of bone quality and bone material, it's very, very relevant in the rare bone diseases, where often we're talking about patients who have tissues which are very poor and crack and fracture easily. And I know you've been working around some treatments for rare bone disease. Could you tell us about it?
Right, so that's true. There's a family of rare bone diseases where the patients have soft bones. The patient advocacy groups around the world are actually called Soft Bones Organization. And so these are patients that fail to completely mineralize their bones. And one of the things we observed, particularly in a rare disease called X-linked hypophosphatemia, in that disease, these patients have bowed legs, knocked knees, bone deformities that are very painful. And the bones, they don't fracture in the traditional sense. When they're loaded, they start bending and buckling. And so the fractures these patients get are called pseudo-fractures. I call them buckling fractures because the bones are soft. So why are the bones soft in these XLH patients? It's because they have a mutation in a gene called FEX that is participating in that regulation of mineralization outside the cell in the extracellular bone matrix like I told you about. And that gene does things to serum phosphate such that the bones don't receive enough phosphate to mineralize properly. It also changes the local extracellular matrix milieu such that there are too many inhibitors of calcification that remain in the bone instead of their being removed. They remain there. So in this disease, this is just one example, the combination of low phosphate availability from the serum and too many inhibitors accumulating in the bone not removed by enzymes renders the bone soft. And when we did our methods on these mouse model of this disease, we found that mineral 3D tiling, cross-fibrillar mineral tessellation, was very defective. I'm the detective. The patients have defective mineralization. So that tiling didn't occur properly in the mouse model. We've since done human patient biopsies, and the tessellation does not occur. It's delayed, it's slow, and it may be never proper. And so this changes then the mechanical ability of the bones to withstand loading, and because of this microscale arrangement, we believe, and thus the bones slowly, upon walking and loading of the children and so on, they slowly bend and deform. They don't suddenly snap fracture. They're slowly soft bending bones because the tessellation tiles are not interlocking with each other. Something simple like that, I think, that's been overlooked for a while.
Is there a possibility that problems with the tessellation then could explain some of the very different clinical pictures we see in sort of standard postmenopausal osteoporosis, where some patients have very low bone mineral density and fall over all the time but never seem to fracture? Other patients have a reasonably good bone mineral density but seem to fracture easily. Is it possible that some problem with the tessellation is sort of part of that issue, and that the way we look at the drugs at the moment we have for that is sort of missing out on that entirely?
So we have to be a little bit careful here, because the kind of work that we're showing, this microscale tessellation pattern, is not a bone density change per se. Bone density is about total mineral content. What we're studying is, given a set amount of total mineral content, that it would be tessellated. You just have less bone in osteoporosis, and thus a lower mineral density at the larger scale. But my guess, and we haven't done osteoporotic bone yet, my guess would be that where there is bone, the tessellation would be fine. And this is all pretty new stuff, so we haven't looked at all the bone diseases, and we're working towards that route. But I think for the moment, we have to say for many diseases, if there's bone, there's probably tessellation everywhere. You just don't have enough bone with the right architecture. The tessellation story goes more to a class of rare diseases called the osteomalacias, and the teeth, odontomalacias, or dentinomalacias. So we will start looking. You know, I hope that not just us, I thought everybody, in the spirit of good science, goes out and tries to find these tessellation patterns. It does require some very expensive, fancy equipment, I must say. So we need some good research funding from our agencies for that. But I don't think it applies to all the bone diseases, just some.
Okay.
Thank you very much for taking the time to talk to us today. It's been a really lovely interview. Thank you.
It's been a pleasure for me too. Thank you.
Thanks very much.
Welcome, listeners. We're delighted to have our next two guests with us here today. We've got Dr. Lidia Zhtynk. Hi.
Hello.
And we've got Laura Vintura. Hi.
Hi.
Thanks for taking the time to speak to us.
Thank you.
How are you finding ECTS 2026?
Oh, nice. Super interesting. Started yesterday with a pre-meeting. Great talks.
And a lot of information to digest, I guess.
Yeah, there's been quite a bit so far about osteogenesis imperfecta, which is one of the things we want to talk to you about today. Tell us your role, Lydia, if you don't mind initially. Why are you here specifically?
So this year I'm representing Osteogenesis Imperfecta Federation Europe, which is an umbrella association for all nation groups of OI. But I'm also like biomedical researcher, so I have like double interest here today.
And you gave a really fantastic talk yesterday about the OIFE. Would you be able to tell us about the organization?
Yes, so OIFE has full members, which are national groups inside the Europe. So we have 20 members. And we have also like associated members who are outside of Europe. So it's from China, Africa, and other countries, America. And then we have associate members which do not have membership of OI people, but they're like supportive organizations or charities. So for example, it's Care for Brittle Bones, which is based in the Netherlands, and also Foundation Ajuce from Spain. And OIF is basically coordinating the national organizations and deals with representative of the patients on the European level in networks for the professionals on different policies and so on.
What are the aims of the organization? What do you hope to deliver?
Yeah, so vision of the OI is about living of active lives for OI kids and also adults and have a nice access to the high quality healthcare and social support.
And if there was something you wanted the listeners to take away that they maybe thought they knew all about OI, but what would patients want listeners to hear that maybe they don't hear about living with OI?
So that first thing is that OI is more than bones. Like we like to say that it's not only the bone but also in the soft tissue, because the collagen is everywhere basically. It's structural protein, so it will affect a lot. The second point maybe it's that OI is very different, and we're talking about differences not between various forms of OI or types, like we say, but also between even children and adults. It's also two completely different disorders. For example, if we're talking about type 1, that in childhood you have a lot of fractures and you're like concentrated on the bone health, but once you're past like your 20s, it's a lot about the pain, fatigue, and the soft tissue problems.
Laura, you're also working in OI research.
Yes, I am. I am doing right now a PhD at Amsterdam UMC in the Netherlands. I do preclinical research. My project focuses on a mouse model for haploinsufficient osteogenesis imperfecta type 1. So for those OI forms caused by deficiency, so quantitative deficiency defects in collagen type 1. Which actually represent a huge proportion of the OI population. And these patients indeed present with bone fragility, but they also suffer from chronic pain, and they also have a lot of extraskeletal manifestations.
Mm-hmm. And what organs are involved in the extraskeletal manifestations? If it's not just bone, what other?
So collagen type 1 is one of the main structural proteins in many tissues, including skin, where it represents the 80-90% of the dermis, but also partly in tendons, in muscles, in dentin. Multiple tissues are involved.
And we could maybe ask Lydia as well, do you think doctors are good at treating the extra articular manifestations or the skin and
Depends who your
tendon
doctors
problems?
are, I guess. Yeah. If it's like a big center who's like has multidisciplinary team, and you're like primary doctor, he knows all about that. Yep. He will send you everywhere to like, I don't know, to check your eyes, to check your teeth, to check your muscles, heart, of course, very important for eye people to also. But if you're somewhere in the smaller city where there's not that good infrastructure, so your GP or your like primary doctor, they will like most likely miss that, unfortunately. So it's also like not about coordination of the specialists only, but also about awareness of the doctors about all this, and it's also about training of the doctors, unfortunately, still.
And is there one particular aspect of those manifestations, Laura, you're looking at?
So we are currently looking at skin and also at muscles and tendons. So we are performing different types of analysis at the structural level mainly. And yeah, why is extremely variable, also due to the different molecular defects causing the different OI types. So,
may I add into that, because we're also looking at the aging process in these mice. So as you know, like there's a lot of fractures, no fractures, and then when later in life with osteoporotic changes, there will be a lot of fractures. So we have like mice which are like plus 42 weeks old, like really old. So we will look also into what happens on the molecular level in the bones and other tissues. So I think it will be, we don't have results yet, but we're like looking into that.
Well, thank you for sharing with us the work so far, and we're going to have to catch up at a future ECTS conference to get an update for the listeners. Thank you very much.
thank you.
Thank you very much.
Thank you.
Listeners, we're delighted to bring back friend of the podcast, Professor Ken Poole from Cambridge University. Welcome back.
Good evening.
It's lovely to see you again here in sunny Catalonia.
Oh, isn't it beautiful?
The debate on Friday was absolutely wonderful. We really enjoyed it. The question was, this house believes that AI can replace physicians in assessing fracture risk.
And you're at a slight advantage because your opponent in the debate unfortunately had to fly off afterwards. So you're here to give us a quick summary of the debate. Tell us the sort of the arguments that came up and the sort of issues involved in the use of AI in this area.
I will, and thanks for inviting me to speak to you again today. It was, obviously the debate is light-hearted, but this year the debate between myself, Ken Poole, and Nick Fuggle, who's professor in Southampton, was just a really, really good opportunity to get out there and look at the cutting edge of where artificial intelligence impacts upon our lives as physicians, the way we do our medicine in osteoporosis. And I think anyone who was there would think that this was a pretty good debate. The quality was good, the entertainment value was pretty high, and there was a clear winner. Nick Fuggle managed to persuade the audience that they weren't ready for AI to replace the physician in the management in the assessment of fracture risk yet.
The issue was specifically whether AI could replace a physician, not in seeing patients necessarily prescribing drugs, but just in identifying, identifying people at risk. You explain what was going on.
Yeah, so in a way, AI can replace the physician was partly where the debate was won and lost, and it seemed to me quite easy to be able to present this simple fact that let's find out what fracture risk assessment is, what the physician does in fracture risk assessment, and then say that AI can replace the physician in doing that. So in my argument, I really put a fence round by using Nick Fuggle's own publications of how we measure fracture risk using FRAX. Then I took it back to simply a large language model, and if you saw, with some entertainment involving my artificial intelligence induced augmented pictures of my family members, and also using Alan Turing, the incredible godfather of artificial intelligence, and actually, if you were looking closely, I made ChatGPT fail the Turing test. I mean, in a very simple way, by asking what ChatGPT thought of the Anthony Gormley statue of Alan Turing right at the start, and then went on just to simply, not in real time, it was all done beforehand, but show that ChatGPT 5 and above was quite easily capable of replacing all the elements of what a physician does in terms of grading fracture risk, asking the right questions in a hypothetical case, which was actually a family case of a vertebral fracture, modifying those risk factors that it produced through FRAX. It made an emulation of the FRAX tool within about 10 seconds, where it really very accurately and could provide its workings. And then it did FRAX Plus to augment with the recent vertebral fracture, and came up with the same result as we would do through conventional means. And then for a sort of coup de grace, showed that this actually did, it could then say that you met the criteria for treatment in the UK. And that was the sort of thrust. The thrust was fairly simple insofar as it was easy to demonstrate that AI can replace the physician in this single task. And then, so there was then a chance for Nick Fuggle, who isn't here, to respond.
Listeners, we did ask Nick Fuggle for comment, but he declined.
Nick had to go home. So shall I go on and summarize?
Yeah, I mean, so I think most people would have grasped that concept that AI, most computing machines could go through the process of calculating fracts and putting the data in and getting a result. So how did Nick, was he asking more, should AI replace a human in doing that, or was it safe for AI to replace a human? What sort of arguments did he put forward?
So, I mean, it's almost impossible to describe Nick's talk with. For anyone who was at the conference or was a member of ECTS, please go to the campus section of ECTS website as soon as it's uploaded and watch the talks, which involved Nick turning me into Peter Pan, him into Marty McFly.
Was it Frack to the Future?
Frack to the Future. He spoke on, he made his there was lots of jokes about his out-of-date photograph. I mean, it was just a real tour de force. He used the AI tools in a brilliant way, and it was just brilliant. But the thrust of his argument really was he discussed the role of AI in medicine in totality, opportunity to improve diagnosis, and find vertebral fractures. He, unlike me, I thought that was a fairly simple thing to do, just to show that you could replace the physician in fracture risk assessment. He broadened it out into different companies like Nanox AI, Ascena, and Euphonia. At one point, if you remember, he had the Euphonia voice doing part of a fracture liaison service interview with a pretend patient, which was extraordinary. And then, but his thrust was that it's critical that AI functions as a tool to support clinicians within a multidisciplinary team and not to replace them. So, and he spent a lot of time talking about regulatory frameworks. I think one place he went really deep was saying, if you don't regulate this, then you can have, and he named a bunch of the companies that had got into a lot of trouble through basically not having good regulation of their products and saying they did things which they clearly couldn't do. And he said people could die. And I think the use of AI as a replacement for clinical judgment is not permissible and carried significant risks. He talked about Therac 25 and things like that. So the future of AI in medicine should be one of augmentation, not automation, to improve patient outcomes safely. Yet you're right that in terms of a debating technique, Nick spent the first 2 or 3 slides just praising me, which was extraordinary. And I was blushing at the front as he talked about my characteristics and said that I was like Peter Pan. And then he moved very quickly to spend about a fifth of his argument on trying to move away from AI can replace and his definitions of is able to replace or
ought to replace or should replace.
Yeah, and that's
replacing.
Do you know? I mean, I wasn't. The thing about this debate was, and I hope you recall my rider right at the beginning, which was, do not do what I am about to do. My employer does not suggest that you do this. And so we had coordinated beforehand. And when he talked about the Wild West out there for AI regulation, you can start shoving these things into all of your large language models, whether it's ChatGPT or Mistral or anything else. They are completely not regulated for this purpose. And you will have seen from the rebuttal that I went almost immediately onto what not to do. Do not upload confidential data. Do not start using these large language models. And then there were the questions from the audience, but people are doing it. They've got 5 or 6 months to wait.
I was stuck on the balcony, so didn't have a microphone, but what I would have maybe asked, and I thought it might have come out a little more, was patient acceptability and what patients think about it, because there seem to be some areas in which patients are hugely keen in AI and ready to embrace AI, but other areas in which they're extremely reluctant to embrace AI. And I sometimes wonder if you were asked the question, you could see a real-life physician, you could see Professor Ken Poole, and he might make an error 1 in 10,000 times, or you could be seen by AI and it might make an error 1 in 10 million times. How many patients would still say, do you know, I'd actually rather see the Peter Pan lookalike Professor Ken Poole, because there's something about human contact?
He was right, though. You haven't aged a bit in the time I've known you.
We were trying to have this debate, and it was just, I couldn't stop laughing. But so you're right. One of the questions asked by Michael McKee, amazing scientist from the States, was, he put it a bit more rudely. He said, I think physicians are wrong 10 to 20% of the time, and I'm much happier with AI being right, whatever fact.
Yeah.
So there was that question. He articulated the question as to, don't you think that people are going to be found to be worse than AI in types of implementations, and that was a question. And then there was another question that did ask, what do the patients feel about this, which I was really pleased with. And if you remember, the answer was to do with Heidi AI, which is the thing we're testing in Addenbrooke's Hospital, Cambridge at the moment, whereby you switch it on, log in, and then you have to get the patient's consent to say, would you mind if this is AI transcribed, and it will then create the note for me, and will leave me some time that I didn't have before, not writing my letter. And in the 6-week trial I've had 0 patients say no to that, which comes back to that thing you said about people are ready to embrace some parts. And didn't someone say, I'm surprised you're not using, the feeling was that people are saying, I'm surprised you're not using AI already, because it's permeated into so many parts of our lives.
Yeah. The debate was absolutely hilarious. The crowd were laughing and laughing and laughing all the way through, especially at Nick Fuggles' finish. It was really wonderful, and I think a lot of fun was had by all. We've spoken to a few people who said that they've been to conferences that don't have debates before, and maybe they're going to go and try and get them.
I think one of the things was, if you recall, one of the ways I demonstrated the nuances of what the ChatGPT 5 and above models were able to do was put my own, shared my own story of having a rib fracture, and I hid from ChatGPT that it was actually a rib fracture because my son had bowled a cricket ball into my ribs, which is a real story. It really happened, and what I used that for was to show that. I mean, my view still is that these things need regulation, and you saw we harmonized on our conclusions, myself and Nick, that do not do this. These things will be available quite soon, but the current environment is they're not regulated, and you can get in a lot of trouble with the wrong answer. But what you saw there was that it very quickly realized that that what it had given me in terms of its fracture risk assessment needed modifying into, you've had a rib fracture, and you did it because of sport, and you did it because of falling off a bike. These were big knocks. You don't need bone pills now. And I actually changed I asked it to imagine that I was an Atletico Madrid football supporter, and said, you need to make this really simple. And because I heard that would be a good joke in Girona, it seemed to get a laugh. But it did, and it said, you've got to tell your son to bowl more slowly. He's only going to get quicker. Yeah. And next time, hit the ball over the boundary while instead of getting it in your skeleton. And it had those humanistic touches.
Yeah, the nice touches, the jokes, the funniness. It sounded quite compassionate.
I think there is a really important aspect to clinical AI at the moment. And that is that it is completely unregulated. Even that slide I showed, where the people writing that paper had to put in an addendum that said that the FDA actually felt that a number of these tools would not actually be subject to regulation because they didn't replace clinician judgment. But actually, even that, where's the point of clinician judgment if the machine is doing all this in the background?
Well, doesn't it come back to this term we use in medicine, medical device? So if you invent a machine, then you have to, that has to be passed as safe, and it has to be regulated before you can connect the machine up to a patient, which is quite correct. Everyone would understand that. Now, to some extent, AI is a calculating machine, but yet people don't recognize it as a machine as such, and that's sort of a grey area legally.
And that was why we included those parts of the talk. And Nick was, one of the things that was that is going to evolve through these as AI gets more competent at different aspects is where exactly is that drawn? Because at the moment, you have with something like the Heidi AI transcription service, you can start asking it. It says, ask me anything you want. So you can say, oh, could you put it together as a referral to an orthopedic surgeon to replace a hip? And it'll do that quite happily from what I've written. But if you said, could you give me the differential? It's got immediate boundaries because it's been built as it is a medical device and it's regulated. If you look down the list of things it can't do, and it'll say, no, I can't do that. And there's so many times with Heidi that you'll get, no, not going to do that, and it's in red, and so there are things that are on the pathway. With ChatGPT, actually in the BBC article this week on the fact that it wasn't very accurate when people put health terms into it, the team behind ChatGPT said, oh, we do interact with doctors, and we realize people use our product for clinical purposes. So where they feel that large language models are going, we don't know. The fact that people are using them all the time, and the hands up, which showed how many people were just using them all the time, and the people in the questions and answers who said, oh, well, I'd rather go to ChatGPT than wait 4 months to see the doctor to ask about this. These are all things which meant, that go back to your question, Richie, that as a conference debate, it could not have been better. It could not have been better now to show people what not to do, to put out some real flags. Don't put real data up to this thing just because you think it's an anonymous or anonymized process. It's not. You can't put patients' data into this and start using it.
Yeah, that's great. If I'd had a microphone, I would have used ChatGPT and asked it what would be an intelligent question to ask Professor Ken Poole and Professor Nick Fuggle at the ECTS debate, but there we are.
Well, I just gave a summary of Nick's talk. Yeah. I was actually running Heidi Health during his part of the debate for the 20 minutes there, and immediately before I walked back up to the microphone, it had summarised his key points.
Wow. It also helped you to form your rebuttal.
Yeah. But so now the mechanics of the vote. When we went in the room, I sort of thought I'd lost already, because immediately when asked the question, for the motion vote went up so fast. If you watch the video back, it got up to 59 before anyone was voting against. So it was a tough job to try and keep those 59 and add to them. Whereas I think Nick did a beautiful job of showing people that quality and safety and regulation is the thing that needs to be done in this situation.
Yeah, well, you're very gracious in defeat, which is possibly one lesson as to how things should be debated in a civilized society.
Oh, well, I mean, like I said to you guys before, if you ever get a chance and you're invited to be either for or against a debate, the space it gives you to think about that question around all your other tasks while you're cycling to work or while you're just on the train or something on the plane, it gives you this chance to really think about it. And I would not give this up for the world, the chance to look at whether AI can replace the physician in this small area of medicine. And I still feel there's if we come back in 5 years' time, we won't even be able to conceive the environment that will be in there. If you remember, I mentioned about the robot that delivered the pizza in Sheffield at the Edge at the Bone Research Society conference, and it just came up to the table, and you think, before you know it, these things have happened.
Well, we considered just using ChatGPT and saying, what would Ken Poole say in an interview if we brought him up here to speak to him at ECTS? But we thought we'd give you the benefit of the doubt and interview the real person. So, listen, thank you very much indeed for coming and speaking to us.
It's my absolute pleasure. Keep up the good work. What you're doing is really valuable.
That's very kind, thank you. And obviously, congratulations to Professor Nick Fuggle for winning the debate. And I think we're going to have to get you on the podcast as well to talk about AI some more. It's a very pressing issue.
Because he's at the Turing Institute and he's absolutely the man to speak to about this.
He'll be gutted that he's missed his BoneUP mug. Thanks, Ken.
Thank you. We're here now with Dr. Judy Keen, Executive Director of the ASBMR. Welcome.
Yeah, thank you very much. I'm very happy to be here.
We've been speaking a lot to you in the last day or two, so it's great to get you with a microphone in your hand and get it on record then.
Yes, thank you.
To begin with, could you tell us a little bit about the ASBMR?
Yeah, so it's the American Society for Bone and Mineral Research. It's been around for about 48 years now. We are the largest society dedicated to research in bone and mineral research in the world. And we've been doing a lot with ECTS and doing some fun programming and looking forward to doing more.
Fantastic. There seems to be quite a close association between ASBMR and ECTS. Yesterday we had the annual debate competition, ASBMR versus ETS, which unfortunately ASBMR won.
Well, no, it was good. We won for the first time since, I believe, 2021. So it's been quite the fun rivalry that we have between the two organizations. And we do it both here at ECTS and at ASBMR. So we'll have a rematch in October.
On this side of the pond, we often find ourselves sort of pushing bone and promoting bone and trying to get more people interested in bone and raise the awareness of osteoporosis and other conditions. Where are you with that? I was going to say in the States, but you represent, of course, all of the Americas. So where are you with that?
Yeah, we are international. We're not just the U.S. But we are also very much interested in promoting awareness of bone diseases and increasing research for musculoskeletal disorders worldwide. We are capitalizing on the recent SABRE decision that has just come out that is the new bone mineral density surrogate marker. And we think that's going to stir a lot of innovation. We've had a lot of interesting talks with people who are interested in now getting more interest in osteoporosis, using that as a way to start really building awareness, because I think we have a great moment and great opportunity right now to do so. And also knowing that it's not just osteoporosis, it's also rare bone diseases that are very important, so we don't want to leave them behind in everything that's happening. But I think that there's been a bit of a shift, and that hopefully this will result in much more awareness, especially among the public. So it's not just us talking to ourselves.
Yeah, there seems to be a lot of really good talks at this conference about osteoporosis, search for new treatments, lots of new technologies which are going to help with searching for those treatments, and quite a lot of talks about rare bone diseases as well, repurposing existing treatments and looking for new treatments. It feels like we're on the up.
Yeah, I agree. I mean, we've had a lot of investors who are interested now and asking about osteoporosis and looking at new treatments and thinking about what's coming out. But also, there's been for the past several years a lot of emphasis on rare bone disease within ASBMR, and a lot of good therapies that are coming out there as well.
And what about collaborations then between science and basic research and practicing clinicians? Because it's something obviously ECTS tries to promote very strongly, and I think we do reasonably well. We could do better. How does that look from the point of view of the ASBMR? Is that easy to do?
No, I think we have a mix, obviously, of researchers and clinicians, and it's always trying to get the two together. How do you translate from the clinic or from the bench to the clinic? And it's something that we really want to try to promote a lot more of, especially now, especially since there's much more interest in what we're doing in the field.
And where is the ASBMR conference this year?
It's in Boston this year in October. So it'll be a good conference. We've got a lot of good things going on. We're trying to do some new things as well. And it's also the 40th anniversary of JBMR. So we're going to have a lot of interesting talks. We're having talks about the important papers and the important things that have come out per decade. So I think it'll be a lot to celebrate and a lot of new things to look forward to.
The JBMR being the Journal of Bone Mineral Research, and in fact, a few episodes ago we spoke to Raj Thakur, and he was extolling the virtues of JBMR as editor and talking about going back and looking at the sort of really important papers decade by decade, sometimes how it wasn't recognized at the time, which the really important paper was.
Right.
But as you say, the SABRE data has now come out, and at the moment certainly we're looking at that as something which will be a big step change in our ability to maybe to get drugs to the market quicker or to get them to patients more quickly, because we'll be able to do more focused and shorter trials, hopefully.
Yes, and I think that's what a lot of people are excited about, that you can actually now have much more innovation in the osteoporosis area because it won't take as long and be as expensive to do it. So that's the hope, and I think that that's starting to happen. There's a lot more investors who are interested in coming to talk to us and to others. I know they're talking to other organizations.
People want to see outcomes, well, patients want to see outcomes quickly, and doctors and scientists, but ultimately investors want to see outcomes quickly as well, don't they? And one of the issues about bone is that it moves fairly slowly, and it sometimes took long trials. So so the SABRE data might well transform that.
Yeah, there was not very much investment from a lot of companies because it took so long. It was so expensive to do those trials. So now the hope is that it's going to be a lot faster.
So listeners, ASBMR in Boston in October. I think the abstracts are still open and close on May the 7th. So if you're thinking about going, get your abstract in.
Please. Yes. Looking forward to seeing you there.
Us too. Thank you, Jude. That's very kind.
Thanks for coming and speaking to us. We'll hopefully see you in Boston.
Yes. Looking forward to it. Very nice meeting you.
Listeners, we hope you've enjoyed the episode brought to you from Girona ECTS 2026. Stay tuned for the next one, which will be out soon.